And I think my talk today will serve as a great complement to Zach Cohane's talk. I was happy to get up a little early and see that just now, and happy to be here from California. What I'd like to do is give a series of examples of AI use in clinical medicine and give a sense of the promise, why this work is so important, and as well talk about some of the risks and challenges to patient safety. So I'd like to start with a clinical example. This is a woman who's 83 years old. She has had an aortic valve replaced in the past and occasionally has difficulty with congestive heart failure. Gets a chest x-ray like this one, which she had about 18 months ago. This was read as normal. She then returned, and you can see she now has this large mass in her right chest. And if you look back carefully at the prior image, you'll see that, in fact, there's a small mass hiding partially behind a rib, nestled among the vessels of the lung. And this unfortunately was missed by the radiologist at the time. And I think it illustrates an important principle and an important use case for these kinds of machine learning technologies. About 3 to 6%, if you look in the literature, of diagnostic imaging studies in medicine have clinically significant errors. So as a radiologist, I think many of us feel an obligation to try to bring these technologies into clinical care to help us avoid errors like this. Briefly, my disclosures: I advise several AI startups, and my department at School of Medicine receives support from a number of industry partners. So just to start, why are we focusing on radiology as an example? This is the health AI industry in the US. These are the approvals, the clearances by the Food and Drug Administration here in the US of AI and machine learning enabled devices over time. And you can see it's really ballooned over the last several years, many of these technologies. And if you break these down by application, the vast majority of them are radiology. And even beyond that, the vast majority are imaging. So this is really a large industry. At our most recent meeting of the Radiological Society of North America, we have about 6 football fields full of trade show space and industry. About one of those football fields was just our AI showcase featuring about 100 companies in this industry. So it's real and it's happening. So what I'd like to do is, in this presentation, is to talk about some of the themes and that you'll see as this industry makes an impact. And those are interdisciplinary collaboration, transformation of healthcare, not just in radiology, but beyond, physician augmentation, intelligent devices, data needs, and safety issues. So we'll spend a fair amount of time at the end talking about threats to safety. So interdisciplinary collaboration just gives me a chance to make a shout out to the center I lead here at Stanford, called the Center for AI in Medicine and Imaging. We are about 140 faculty at Stanford across 20 departments who are all focused on issues of machine learning in medicine, not just in imaging, but across the board, including EHR data, genomics, and other applications. So a lot of good collaborations resulted in the Stanford work that you'll see today. All right, let's talk about how AI can transform healthcare outside the radiology department. First example I'd like to give is, this is the work of David Ouyang, who is one of our cardiology fellows. This was published in Nature. And this has to do with ejection fraction, which is a key variable used in cardiac care. He built a model that processed the video of an echocardiogram and was able to calculate accurately the ejection fraction to an area under the ROC curve of about 0.96. This was using semantic segmentations with dilated convolutions together with a spatiotemporal model. And it was trained at Stanford and externally validated using data from another hospital. So it showed you could predict, it also showed you could predict other human characteristics like gender, age, height, and weight that we didn't really realize was possible to do from these kinds of images. At least humans had not done that in the past. And we'll return to that a little later in the talk. Another interesting example relates to what I'll call population health screening or opportunistic screening. So this study on the left is what we call a gated CT study. The images are acquired at the same point in the cardiac cycle. So the heart is at the same point in contracting, and that allows us to freeze the heart. And you can see there that the white dots are the calcium in the coronary arteries, and that helps us, the level of calcium in the coronary arteries allows us to assess cardiovascular risk. The cost of this test can be around $2,000. On the right, we have the same patient who's imaged routinely. We might obtain this test for someone who's had trauma or may have pneumonia or following a lung nodule. And we don't typically gate the heart in that type of a test. It's just a routine CT of the chest. And so what we did is built an AI model that can still assess cardiovascular risk, even though the calcium in the arteries you can see there is smudged in the yellow circles. So this gives us an opportunity at no additional cost of screening hundreds of thousands of patients who receive these CTs every year for their cardiovascular risk. So this has now been validated at 4 disparate sites as well. This is a encoder-decoder architecture with a 50-layer ResNet pre-trained on ImageNet, and had some skip connections similar to a UNet. This is the work of Matt Lundgren with our Andrew Ng and our collaborations in computer science. Expert level chest radiograph interpretation. So this was a 121-layer DenseNet architecture built to detect 14 common abnormalities on a chest radiograph. And you can see that the algorithm is better than the expert radiologists for one abnormality. The radiologists were better in 3, and they were indistinguishable in the others. So we're getting expert level performance out of this algorithm. Here's just some more data specifically on nodules and consolidation. And you can see the purple diamonds here are the algorithm. The open diamonds are the trainee radiologists. The closed diamonds are the board certified radiologists. And you can see, again, for all these measures of accuracy, we're getting expert level performance. One of the ways that we're planning to use this algorithm is to prioritize our work lists so that the cases that are more likely to have abnormalities will come to the top of the list and be interpreted first. We're also in the midst of a trial, a multi-institutional trial at 7 emergency department sites in the Intermountain Healthcare Network in the Western United States. They have been using an algorithm to detect pneumonia in patients in their emergency departments that uses these data points listed here: chest X-ray findings, vital signs, lab values, nursing assessment, age, and chief complaint. And the chest X-ray findings actually are using some natural language processing of the radiology report. That's the way it had been used, and they have this, they've validated this algorithm. What we're now trying is using the pneumonia algorithm I just showed you to replace the radiologist interpretation. This particular data item took the longest to come back by far because had to wait for the image to rise in the queue, for the radiologist to interpret the image, and then for the report to be processed and to become a data point for this algorithm. Instead, now we can process the images directly with the machine learning algorithm and use that as part of this overall algorithm to triage patients in the emergency department. Another way in which an algorithm like this can affect care outside the radiology department is global health. About 1/3 of the world is underserved by radiologists. And so we trained the algorithm to use not just digital radiographs, something that we would find at a hospital like Stanford, but actual photographs of film, which is the way that radiography is often performed in those underserved areas. And you can see there, if you take your smartphone, you take the photograph, you upload this image, and it can provide a bar graph essentially of the different differential diagnosis possibilities. So that's some of the ways in which these algorithms can affect care throughout the health system, even though we're talking today about radiology. The next theme I want to cover is augmentation. So what we've talked about so far is comparing the algorithm to the human. And that's probably not the right way to think about it, because these algorithms will be used in collaboration between the computer and the human. So here's an example. This is from Kristen Yam's lab. She's one of our neuroradiologists. This is an algorithm to detect vascular aneurysms in the brain. And so rather than compare the algorithm to the human, her group compared humans augmented and unaugmented. So the orange dots here are humans interpreting these studies without the algorithm, the blue dots with the algorithm, and you can see that for sensitivity and specificity, the algorithm is causing improvements in those for the most part across the board for radiologists and neurosurgeons. This is another algorithm. This was, again, a collaboration between several investigators in our medical school and in computer science. And this is this is detecting ligamentous abnormalities in MRI of the knee. This is an ROC plot, so you want to be up and to the left, high sensitivity, high specificity. That area is magnified here in the center. And you can see the red X's are unassisted, the blue are assisted. And this is a three-dimensional, 3 planes of images, each pass through AlexNet to obtain a feature tensor, and then global average pooling and max pooling to obtain a 256 length feature vector that was run through a fully connected layer. And so this provides, as you can see, the assistance that this provides typically brings people, radiologists and orthopedic surgeons, up and to the left or along the ROC curve towards greater specificity, which is also valuable. So that's just a theme of augmentation, that these algorithms can augment even expert radiologists in interpreting these images. The next theme I want to mention has to do with intelligent devices. So this is the work of Greg Zaharchuk's lab, another one of our neuroradiologists. On the left here, we have a pre-contrast MR of the brain. For some of our perfusion studies, we actually obtain a 10% contrast dose study, which you're seeing in the middle here. And then the full contrast dose of gadolinium contrast is here on the right. What he did is used one of these convolutional neural networks to produce a synthetic image of what the full contrast dose would look like from just the 10% dose. And then did some interesting studies to show that the images were thought to be of similar quality, and then doing diagnostic studies with radiologists to see whether the same conditions could be diagnosed from these synthetic images. So, and those kinds of comparisons are extremely important because these methods can sometimes either eliminate abnormalities or hallucinate abnormalities. So those kinds of reader studies really are important to do to validate these techniques. Here's another one, same lab, different imaging modality. So this is a MRI. This is a PET MR. So we have the MRI image. We have a low dose PET. So this is just 1% of the counts coming off of our positron emission tomography scanner. And then same principle. Here's the synthetic image. Here's the full dose. And you can see these are quite comparable. And again, a study showing comparable accuracy. So this is a kind of method that can reduce radiation dose, can reduce contrast dose, can reduce imaging times to improve the efficiency and productivity use of these expensive devices. The other way in which these methods will likely change the imaging business is through less expensive devices. So we're now seeing this is a portable MR unit that you can roll around the hospital. Here we have a ultrasound transducer that can be connected directly to a smartphone that's now widely used. The images that these devices would typically produce are not of particularly high quality, but we can use some of these machine learning methods to enhance the images to a point where they become diagnostic images that we're expecting to interpret. Here you can just see the again how this imaging device works. So that's intelligent devices. And now I'd like to move on and talk about data needs. So like any other area of machine learning, data is where we spend a lot of our time. How do we acquire the right data? Curating, cleaning the data, making it suitable, making it AI ready, machine learning ready. And the first principle about data I want to share with you is some work that we did in collaboration with some of the ethicists here at Stanford. This is the work of David Larson primarily. And that is just some principles for ethical use and sharing of clinical imaging data for AI. And our conclusion was, after the clinical data are used to provide care, the primary purpose for acquiring the data is fulfilled. And at that point, clinical data should be treated as a form of public good to be used for the benefit of future patients. So data are not owned in the traditional sense. Instead, the stakeholders who control the data or who manage the data have an obligation to ensure that the data is used to benefit future patients and society. And so there really are a number of labs now that have taken this to heart and have released publicly imaging data sets. This got started from Ron Summers' lab at the National Institutes of Health here, who released 30,000 chest x-rays, followed that up with 32,000 CT images. The folks at NYU have released a very nice MRI reconstruction data set for knees. We released, in conjunction with the Mimic group at MIT, chest x-rays and a labeling method. So we now have over 600,000 labeled chest x-ray images available for machine learning experiments. There are ophthalmology data sets, pathology, histology data sets as well, but still a relatively small number. Here at Stanford, we've now released 15 AI-ready data sets, which I think last time I checked was still more than the rest of the world combined, but perhaps I hope the world is now catching up. So 1,400 knee MRs, 10,000 musculoskeletal radiographs, again, each with labels. And often we release these data and then employ them in public data science challenges, either through the RSNA or just through Stanford, to really engage the field and get people to focus on optimizing the diagnostic accuracy of some of the machine learning methods applied to these data sets. But this kind of a release program that I'm talking about now, and all of the work that everyone has done to release these data sets, really isn't enough. If you look, and this is some work that we did published in JAMA last year, if you look at where the data is coming from, it's at least in the US, this is all the cohorts, US patient cohorts used to train clinical machine learning algorithms by state. And you can see the majority of these are coming from just 3 states. And of course, the demographics of those states is not necessarily, and certainly isn't, representative of many of the other states. And I think you could make a similar pattern for other countries. So it's really imperative for us to broaden the sources of data. And so one of the other answers to that is this project many of us are involved in, called the Medical Imaging and Data Resource Center. It's a federally funded project here in the United States. It's our imaging community's response to the COVID-19 pandemic. And we are pulling together very large, trustworthy data sets of images obtained on patients who have COVID-19 and who don't have COVID-19, so that we can develop machine learning algorithms to detect, characterize COVID-19, make predictions, risk predictions, and find complications of COVID-19. And as you can see here, because we've collected data from this wide variety of sites across the country, the demographics of our data mirrors that of the 2020 census here in the United States. And this data has now been used for many important purposes. We've ingested over 62,000 imaging studies, predominantly chest radiographs and chest CT. About 50,000 of those studies are currently undergoing our quality assessment. So that has to do with assessment for diversity. We also sequester about 20% of the data, so that are never shown to the public. And that's used as a private test set that can be then subsampled each time someone comes with a particular use case that they'd like to have validated. About 13,000 of those studies have been made publicly available. High quality, diverse, representative imaging studies. And more than 24 algorithms have now been produced based on this data set for diagnosis, prognosis, severity, severity characterization, or prediction of things like length of hospital stay. So that's one response to the data problem. Another response has to do with federated or distributed learning. So many of you may be familiar with this, but your typical experiment starts with data from a single organization. And then if we would like to have a generalizable algorithm, we really ought to train it on more than one organization's data. And so what we're finding is that sorry, my slide is not doing what it's supposed to be doing here, but normally what we would do is take the algorithm and pool data from 6 different organizations and then run the algorithm on the pooled data. But instead, what we do with federated learning is we actually move the algorithm or the parameters of the algorithm around among multiple organizations. So rather than pooling the data in a single data set, we sequentially train the algorithm. That's a very nice solution. It enables each organization to retain the data and mitigates privacy concerns. There is some work to show that, and this is work from Daniel Rubin and Jayshree Kalpathy-Kramer's labs, that shows here on as the heterogeneity of the data increases, the accuracy of these algorithms that are trained using this distributed approach degrades. And so we have to be very careful if we're going to use this approach that the labeling methods, for example, are constant across organizations. And in fact, the data provenance, the type of data that we're using is similar. And that's often not the case. So these are really important methods. Similar methods like synthetic data and differential privacy also may play a role here, but I think it's fairly early to say that these can replace traditional pooling approach. And I think for quite a while, we'll see both pooling and these kinds of distributed approaches to machine learning. The other important principle is that we're really very fortunate in radiology and other imaging disciplines in that for each imaging study, a human radiologist produces a narrative report that describes what the images show at the time of that's done for clinical care. So we have this very nice paired data set of images and text, and the text can be used to rapidly produce large numbers of labels. So we can get very large, not perfectly labeled, some you could call it noisy labels, but good enough for training these machine learning algorithms. And these methods have really gone through the same kind of revolution that the computer vision methods have gone through over the past several years. These are some of the methods started with word2vec and these other vector-based methods. And now we're into these what are called transformer models. And all of them are really based on the principle of this linguist, J.R. Firth, who said in 1957, "You shall know a word by the company it keeps." Fundamentally, how these work is that you take a large corpus of text, very, very, very large. So many of these are trained on, for example, if outside of medicine, are trained on the entire corpus of Wikipedia, for example. And you play a little game, an artificial task with the computer, and you say, look, we're going to obscure this word, try to predict what the missing word is. And you just kind of step through the body of text, keep marching through, each time asking the algorithm over and over again to make these predictions. And it turns out that the algorithm, the neural network that's encoding this information, learns a lot about language, context, and word usage by trying to perform that task. So we've done similar kinds of things with very large corpora of radiology reports and pre-trained these kinds of transformer models on this form of clinical text. And then this is the work actually out of my laboratory published at NeurIPS this year. We then use these models to extract information, in this case, knowledge graphs from the clinical reports. So we're extracting both entities here, like an opacity, an infection, pneumothorax, as well as the relations between them. So here we have an opacity that is located in the right lower lobe. It's increased and it is concerning for infection. So that kind of information, it's essentially a small knowledge graph, and it can be used very effectively as a way of labeling large numbers of training data. And here you just see some results from that, just showing that, number one, the coverage is quite high. So we were able to annotate almost 90% of the sentences using a very simple information model, just that included anatomy, observations, and those relations. And you can see that the accuracy that we achieved was quite close to human annotators in most cases. And that's, again, doesn't need to be perfect, because these kinds of weakly labeled or uncertain labels are still very helpful for training these kinds of models. So I've spent the first 25 minutes or so here talking about how amazing some of these machine learning methods are. And now I'd like to talk to you about some of the risks to safety, and start with a kind of cautionary tale about bias that relates to the first algorithm that we built out of our center. This is now several years ago. I think this was the first machine learning model to be published in our journal Radiology. So this is the case of bone age. So we may have a child who's suspected of having some form of developmental delay, and we want to compare their physiologic age to their chronologic age. And one way of assessing their physiologic age is by taking an X-ray of the hand and looking at the pattern of ossification of the cartilage in the wrist and hand bones over time. The reference data for this come from a book called Grulich and Pyle. And so what a human would do is page through the book, find an image that looks about like the image that they're looking at for this patient, and say, okay, that's a 7.5-year-old, therefore this patient is probably 7.5. The reference data for that book came from 300 white children who grew up in Cleveland in the 1950s. We thought this was a good area to start with. It's one single planar image, and the output is a single quantity, the patient's age. So a nice way to get started in machine learning. As I said, was published in Radiology, and you can see the results here. So this is the difference from ground truth. So lower bars are better. And we had 4 expert radiologists versus the machine, which is the lighter bars here. And you can see the machine was significantly better than 2 and about the same as 2 others. So again, human level performance we're getting from this algorithm. And we looked at some of the heat maps, and we'll talk about some of the weaknesses of these in a minute. But nevertheless, they're showing us that the algorithm is looking at things like the wrist bones and the joints here, which is exactly where a human radiologist would look to find where, the level of bone development. Now, we actually went on to study this, that was using data from just 2 institutions. We wanted to study this in a prospective clinical trial, which is, the way that I think many of these methods do need to be assessed. There were over 1,900 studies in this trial, interpreted by 93 radiologists from 3 healthcare centers. And again, we showed that there was, first of all, it was quicker to interpret a study when you had this assistance. And second, that there was an improvement, just as we've seen with some of the other algorithms, with AI assistance at the centers. So that's a great success. But there's a hitch. And that is that there are these racial differences in growth patterns associated with the development of bones across ethnic groups. And so we had built a model based on the reference data for white children, but it was unclear whether this would work for underrepresented groups. And so, it's an interesting question at this point, and really gets to, why is that difference the case? If that difference is due to, let's say, societal factors and access to care or nutrition or something else, then really we ought to develop a different, we have to fix those societal issues. On the other hand, if it's just a genetic difference, it might be appropriate for us to develop, and we can now do this, develop different models for different groups, or one model that will have a sufficient training data for each group. So those are really important questions, and we'll return to that in a moment. Just to give you a really concrete example of this kind of thing. This is work just published in Nature Medicine just very recently about underdiagnosis bias that these algorithms can have. This is chest radiographs, and looking at, so this is the false positive rate for no finding. So essentially, this is a false negative rate because it's a false positive for a negative. It ends up being a false negative rate. So this is going to give you a sense of how much underdiagnosis there is, that we're maybe missing abnormalities. And so you can see in women, in young patients, in Black and Hispanic patients, and in Medicaid, so lower income patients, these are the patients who are having, for whom the algorithm has less sensitivity. So you can see that if you're not very careful about the kinds of training data that are used, you can get into these, you can build machine learning models that have these inherent biases. And this is a very good article I highly recommend on ethical machine learning in healthcare. And I won't go through this in detail, but I think the main point that I take away, at least as it applies to that bone age model that I just mentioned, is that if you're thinking about bias as you develop or after you've developed the algorithm and how to mitigate bias, it's probably too late. And that you really want to think about it early on when you're considering what problem are we solving? Are we trying to build an algorithm that mimics Grulich and Pyle, which has these data on white patients, or are we going to try to collect data that has sufficient data to train an accurate model on all of the patient groups, including underserved groups? Very important principle. Okay, another key problem with these models is just generalizability. Many examples of algorithms that have been trained on data from one health system and don't apply well to data from another health system. And that really also gets to the regulatory approval process. And often those approvals occur on data sets that are quite small or from single institutions. And so just because the algorithm is approved doesn't necessarily mean that it will work on data from your own hospital. And a lot of due diligence is needed for these models. This just illustrates how confounders can play a role also in generalizability. So this is some nice work from Eric Orman's lab. They had built an algorithm to detect pneumonia, and they trained it on data from two institutions. One institution here on the left, many of the patients were in the ICU and were fairly sick, as you can see. And the data from the other institution, these tended to be outpatients who were suspicious of having pneumonia, but most of them ended up having bronchitis or some other malady. And so what the algorithm ended up doing was, rather than really looking at the information in the lungs, was looking at the markers on the image, because these two hospitals use slightly different markers. Here's a L with a circle around it. Here's a backwards L. And so that ended up being the information that is used. And that obviously will affect generalizability if your algorithm's training on the wrong thing. The other major problem that we have in these machine learning methods has to do with reliability over time. And I think Zach mentioned this in his talk as well. So you may have a new imaging device which has a different noise profile. We may have a new patient population. So a new clinic comes online. So now we're seeing patients with different demographic mix, for example, or disease mix. New diseases like COVID can come along and can change the way an algorithm behaves. And so we made the case in this article, this again, primarily authored by David Larson, on regulatory frameworks that really, as we approve these, just as we have post-market surveillance of drugs, for example, we need something similar. Actually, each organization that implements these models really needs to view this through a quality improvement lens, that you need to view it holistically, not just as an AI algorithm, but there's a whole workflow around that algorithm that may change. And there's a need for ongoing monitoring of the behavior of that algorithm to assure that an algorithm that works on day 1 is going to continue to work on day 100. Here's an example of just that kind of shift. So this is work this is an algorithm that's segmenting out the left ventricle on an MR of the heart. And you can see here how the algorithm works very nice on data from manufacturer number 1, where it was trained. But if you try to apply it to manufacturer number 2, all of a sudden the algorithm, its performance breaks down significantly. So important to consider and to monitor algorithms over time. This is the article that actually was just mentioned in one of the questions earlier to Zach's talk, where we have here's a dermatology lesion. rated by the algorithm as benign. Then we add some adversarially created noise, which really isn't perceptible to the human eye, but now this case is rated as malignant. I think this is really important because it illustrates how sensitive or brittle really these algorithms are to changes in the data and the noise patterns. I'm not sure this is practically a real risk in that most of the networks that use these kinds of algorithms are quite secure, and that anyone who would want to penetrate the network is probably more likely to install some ransomware or something of that nature rather than to try to modify the diagnosis of a skin lesion in somebody who they've never met. But I suppose it could happen. We need to continue to monitor the robustness of these algorithms. Now getting to this issue of explainability and trustworthiness. This is a really nice article from J. Srikalpathy Kramer's lab at Harvard, looking at these typical methods, the heat maps that I've been showing you, and whether they're really reliably show us where the algorithm, what data the algorithm is using to make its conclusions. Do these algorithms localize well? Do they vary depending on how the machine learning model is, the initial weights are randomized? Is it even repeatable with the same model? Is it reproducible across models? And here you can see these are 8 different methods for creating these heat maps. This is GradCam, which is a very common one. It happened to perform pretty well on this pneumothorax example, but many of these you can see are really not doing very well. This is comparing DenseNet and Inception v3, 2 commonly used models. So very important for us to recognize that these heat maps are not necessarily reliable explanation methods. On the other hand, I agree with the points made in the talk earlier, which is that explainability in some ways is a secondary issue. This is a wonderful article, just came out in Journal of Clinical Epidemiology, that talks about the fact that explainability in machine learning and healthcare is not necessarily intrinsic, but is instead instrumental to achieving the greater imperatives, which are performance and trust. And the way we deal with the way that we measure those things are the same way we measure, the fact that Tylenol works, which again, we don't really know how it works. We just know that it does. And that's through things like clinical trials. This is from the University of Toronto. I want to leave you with a mystery, and this is a study called Reading Race. It has not been published in a peer-reviewed journal yet, but it's been published on arXiv and creating quite a stir because I don't think anyone fully understands this data to date. So what was done, this was an all-star team of machine learning workers in radiology. They looked at 9 radiology imaging data sets and the ResNet 34 model. And they found that they were able to discern, the model, the algorithm was able to learn how to discern the race of the patient in the image, something that humans are not able to do. And they thought, well, maybe this is overfitting. Here you're seeing the AUC. These are very high areas under the ROC curve. Maybe this was just overfitting. Let's do some external validation. They did, I'm not showing the data, but they externally validated on 6 other data sets. They found similar information. And then they started investigating where is this information coming from that humans aren't able to see. And so they added some noise, they added blurring, and found very similar accuracy results. They tried occluding various parts of the image, particularly those areas of high saliency, and got, again, very similar results. They did patch-based training, looking at different parts of the image, and again, were able to get very high accuracy. They ran the images through a high pass and a low pass filter. Here you can see high pass filter of 100 has very little information that a human would glean, and yet still getting fairly high here accuracy in the AUROC of 0.9. So I don't think anyone fully understands what's happening here. I think many of us, when we first saw this, the interpretation was, well, this must be some kind of methodologic error. But in fact, this is, if you read the paper, it's very solid work and really a conundrum that none of us fully understand. And then this is a late breaking slide that I added because this was discussed in the Q&A earlier. And this is about why radiologists learn MRI physics. So if you think about the fact that humans are going to be working in partnership with these models for quite some time, when MR first came out in radiology, there are some very important physical principles that come to bear. And radiologists incorporated some training about MR physics into our residencies. And I think that was very important because if you look at, for example, this image, this little spot here looks for all the world like a mass in the liver. But if you know about the phase encoding direction and that pulsatile flow in the aorta can cause ghosts, you know that this is actually a phase artifact. Same here, this little area of high signal in the posterior aspect of the brain here actually is a phase artifact of this. This is the sigmoid venous sinus. It's just a shadow. So knowing something about how MR scanners work, not entirely everything about the physics, but knowing something about the physics is very important for radiologists because we understand how these devices might fail us. And I think the same will be true for AI. We will be working in partnership with these algorithms for a long time to come. They're likely to transform the way that radiology is practiced. And we are going to need to learn more. Radiologists are going to need to incorporate training on AI algorithms and heat maps and all of those things to understand the shortcomings so that we can partner with them and know when they may be leading us astray. So with that, I think it's an incredibly exciting time in our field. There's lots to be encouraged by. I'm grateful to play a role in this kind of transformation of medicine and imaging. I hope I've given you a sense of the immense possibilities as well as some of the risks and challenges. And I look forward to your questions. Thank you.
Thank you very much, Professor Langlotz, for a very, very interesting talk indeed. There are several questions here, so I'm going to read them out. This one is from Aaron Margolis, who asks, where can we find the publicly released data and challenges?
Yeah, great question. So the released data sets, if you go to our website, aimi.stanford.edu, you will find the data sets that we have released. And I believe if you look at Ron Summers' lab at NIH, you'll see those data sets. But I will tell you that we have just been funded by the Gordon Betty Moore Foundation to do a survey. Look at a couple of things. One, why don't more organizations share their data? And so there you get into things like risk aversion and privacy issues and difficulties in de-identifying data, which is not as much an issue in images as it is with text data. We're still working through text ourselves and how we would release that publicly. And then to apply, as well, look at data sets themselves and what constitutes a good AI-ready data set that someone would want to release. And then taking those frameworks, organizational and data frameworks, and applying them to all of the data sets that are available out there and creating a kind of unified directory, if you will. So that doesn't exist today, unfortunately, but we're hoping in another year or so, we'll have something that will make it easier for people to find all the data sets that are out there.
Great, thank you. Another one from Horvey Kukina. Thank you for your wonderful talk. Have you tried to implement any deep reinforcement learning algorithms such as the DeepQ network?
Yeah, so that is certainly a very interesting area. We have not, I don't believe, at least in the imaging area, we have not, because those methods tend to work better for repetitive decisions over time. And it's not something that we typically do in imaging. The kinds of decisions we make are overwhelmed by the pixels that we receive with each imaging study. There's some work going on comparing current to prior. I think that's very important. How have things changed? I think that's more relevant, for example, in test ordering behavior and other kinds of repetitive decisions when we're working with EHR data, but a very interesting area.
Great. Here's another one, Dominic Connor. Interested in the ethnicity detection, something I thought they did anyway, given that different groups have physiological differences. Has that been accounted for?
Yeah, so, well, I don't think anyone tried it until that paper I showed you, the reading race paper, until recently, just because, at least for diagnostic imaging, no one believed that there was any signal there, because humans have been looking at these images for decades and decades and haven't seen anything like that. Yes, I think there are physiologic differences, but again, like the bone age example, we need to be careful that are those truly intrinsic, or are those also due to other factors that we should address, not through a machine learning algorithm, but through societal changes. So I think we need to be attuned to that.
And here's one from Dominic. Isn't it impractical to expect physicians to understand the underlying physics of these devices, given that the technology will become more and more complex in the future?
Yeah, well, it's a good question. MR devices are pretty complex too. And I think there's just no way around it, that we are going to have to learn, and it may be more empirically, what are the most important shortcomings and how do those tend to occur? And those shortcomings may change over time as the technology changes and evolves, but I don't see any way around training medical students and residents of all kinds, including radiology residents, to learn about the shortcomings of these methods. So I don't see a great solution there, other than just hard work. So, our MR physics training is substantial in radiology residency now, and I think that's appropriate. And I think that training on machine learning is going to have to become a part of our training, and is going to have to grow over time as well.
And here's one from Bin Yu. Thanks for a great talk. How much documentation on the data collection process there is in the Stanford medical imaging database? I believe the bar should be much higher, or there should be much more info than, say, ImageNet.
I'm sorry, could you repeat that? I missed the last part of that.
Yeah, so I believe the bar should be much higher, or there should be much more info than, say, ImageNet. This is in the chat, not the Q&A. There's a question that came in the chat instead. I'm
not sure what the bar much higher means exactly, but let me put it this way. There are a couple ways to think about that question. One is that there are many questions that we can answer with just images, but there are many more questions that we need, not just images, but other data from the patient's record. So we will need to build models that incorporate not just the imaging data, but other forms of data. So most of the data sets that we've released have been just imaging data with a little bit of additional data about demographics and insurance status and things like that. But I think increasingly in the MIDRIC project, you'll see is releasing quite a bit of additional data, including the images. And we're actually working with other national projects. So there's another project called N3C that has aggregated over 1 million patients' records who have COVID. And that data can be married to our images. And so then we can develop multimodal models. So that's kind of one way to answer the question. The high bar versus ImageNet, I guess what the thought that triggers is that early on, we initialized these machine learning models randomly. And then we realized, well, if we pre-trained it on ImageNet, it learns quite a bit about edge detection and other things that's very useful. And so that improves our accuracy quite a bit. And now we're realizing that ImageNet is really not as good as some other, I didn't have time to show you today, but some other self-supervised learning methods that we are developing in our lab. So one of my grad students, Yuhao Zhang, developed something called contrastive learning method. So we build an encoder to encode the image information. We build an encoder to encode the text information from a radiology report. And then we feed it positive and negative examples of either matched images and reports or unmatched images and reports, and ask it to distinguish between a true and a false pairing. And just in doing that, that image encoder learns a great deal about how to interpret medical images. Then we can take that encoder and use that as a form of pre-training, and then fine tune that encoder on whatever specific task we may need. And that actually works even better than ImageNet pre-training and other forms of pre-training that have come before. So I'm not sure if I answered that question. I answered it in a couple of different ways, but I think that's how I would respond.
So here's one from Stefan Kolik Martinez de Azagra. Thanks for a great talk. How widespread and important do you think differential privacy methods such as DPSGD will become in medical imaging in practice?
Yeah, the short answer is I don't know yet. If you look at these issues, so for example, the differential privacy efforts that I have seen have answered some fairly basic statistics, create data sets or allow you to answer questions that are fairly basic. But I have not seen methods that work well for some of the more complicated methods and for the volume of data requests that we need to make for our machine learning experiments. We could get there. Synthetic data, it's the same. So if you can now create a synthetic data set that has many of the characteristics of the original data set, but that is private or doesn't expose any patient information. The problem there is, again, it does do well with some, fairly basic descriptive statistics on the broad population. But if you look at the rarer diseases or the underserved populations, the answers for those groups tend not to be preserved as well. So I guess I'm not saying that I don't think these methods will work, but I do think that they are going to require a lot more research before they're able to truly replace a large swath of what we would do with a pooled data set. So I'm kind of encouraged by them, and I'm very glad there's work going on, but I don't think they're quite ready yet, at least for the practical use that the kinds of algorithms that we'd like to build today.
And here's one from Suri Parthasarathi. Thanks for your wonderful talk. In cases where the availability of original data is not possible due to privacy or regulation, what is your recommendation of preparing synthetic data for medical imaging?
Yeah, I think I answered this a little bit in my previous response, and that is that I think that these synthetic data sets are useful to answer some basic questions, but I'm not sure that they work well for all questions and all groups. So I don't have a great answer other than I do think, as a matter of principle, that most patients actually want us to use their data for these forms of research. So I think that we need to find ways to release the data, to make it more widely available. I think that's synthetic data. And as I said, I think research on that is valuable, but I'm not sure that it'll ever replace the need for real data. And I think there are ways to make real data available that is private and that actually is consistent with the needs and desires of the patients from whom that data was created.
So I have one question which relates to the issue of comparing with humans, and it'd be great to get your expertise on this as an expert in radiology. So many of the cases that you show, there's a comparison with the human, and typically the machine outperforms the human in many cases. Now, you also show this interesting example from Cohen's paper in Science, where there is a self-assessment of the neural network that tells about its confidence in its own prediction. And as is clearly shown, that is sometimes completely nonsensical. Now, in many cases, humans, of course, can have overly confident assessments of their own abilities. We have plenty of examples, but there are also cases for which the human is pretty good at understanding its limitations. So in terms of testing against a human, should one also test the trust level? And how would that compare? Because if I were to use, an AI or a doctor, and if somebody can statistically show me that the doctor says that I don't have cancer, well, then I can sleep soundly. Whereas if somebody shows me this example from Cohen's paper, and I realize that the very confident prediction that I don't have cancer may not make me sleep so soundly. So I'm wondering if you can comment on these issues of trust.
Yeah. Well, first of all, I think it's really helpful for both humans and algorithms to try to quantify the level of confidence that they have in their conclusions and to work with patients directly on that for this sort of collaborative decision making. In terms of trustworthiness, as I said in the talk, I think it's less about the heat map or something of that nature, and more about clinical trials and other studies that show that an algorithm works in a given clinical situation. So I don't. I guess what I would say is that trustworthiness is that higher goal. Having said that, we're also seeing instances where the human trusts the algorithm too much. So automation bias, in a sense, that because the AI said this, I'm going to believe the AI. And I think we have a lot of work to do on the user interface, if you will, the user interaction, human-computer interaction component of this, particularly as the number of algorithms grows. So if we're just talking about an algorithm that detects pneumonia, for example, okay, that's one thing that puts a marker on the pneumonia. I can assess the confidence there. Well, what about, as we start to build in nodules, pneumothorax, pleural effusion, cardiomegaly, all these different, and we have 12 or 14 different abnormalities, all of which may be marked on an image, and we don't have a good sense of confidence. How are we going to present that to the user in a way that doesn't end up being more confusing than helpful? And I think that that's a real open question, both just the sheer number of different alerts that may be happening, and also this sort of how do we communicate the algorithm's confidence, and how do we make sure that that confidence is calibrated so that the human can interpret it in the right way. So those are all really important challenges, and I think those are active research questions that we have to tackle today, because I don't think people have yet thought enough about those problems.
Great, thank you. I think we can do one more question before we go into the next speaker. So this is David Taylor. I agree with your comment that patients want their data to be used for research once they understand what we are talking about. Can you cite any specific evidence that patients understand the need for AI and that are keen to consent for their data to be used?
Yeah, I can't give you I'd be happy to give you the reference, but there have been a couple of fairly good patient surveys in terms of what patients want. And if you ask them, under the assumption that your data will be kept, your privacy will not be violated, and the data will be used responsibly by scientists to better human knowledge and create technologies that are going to help other patients, would you want your data to be used? And the answer is overwhelmingly yes. So I think that we are in a good place when it comes to data sharing. That's true not just for AI. I don't know that I can cite you evidence that patients understand AI or think AI is a great thing. And I think that's something that still has to play out. And it does. I think that what's happening outside of healthcare tends to color people's opinions of it probably in a negative way today. So we have some work to do there. But I think the encouraging thing is that patients generally do want us to try to use these technologies to better their health. And I think we're definitely on the road to do that.
Great. Thanks very much for a very, very interesting talk indeed, Professor Langlotz. Thanks so much for being with us today. Really, really appreciate it.
Thank you. It was my pleasure.